like monocytes. They have multiple nuclei. They have long processes pseudopodia, have almost a ruffled appearance. Remember, osteoclasts are derived from the same stem cell as monocytes are. And so it's kind of like a plasma cell and a lymphocyte. The functional monocyte looks quite different. Okay, leukemia is a type of cancer in which there are too many white blood cells, and yet these individuals often will first demonstrate with a fever because these are immature white blood cells and they're not functional. So even though we have lots of white blood cells, the person has a difficult time fighting infections, and they crowd out the red blood cells. So they show up with weakness and being tired because of not enough red blood cells. In children, leukemia is one of the most, what's the word I want, not easily, has the highest cure rate because so much money and research has been done with leukemia over the last 50 years. Hospitals like St. Jude's and so on, and so I think it's almost an 85% cure rate with childhood leukemia. In adults, however, it's a different story. Okay, my best friend's mom died about 10 years ago. She was in her early 60s, but she developed a pediatric form of leukemia. And she just had a fever for a week or so and finally went into the doctor, expected to be told it was a virus or being given some antibiotics, and they did a blood test and came back with a diagnosis of leukemia. That was in March, about 10 years ago, and she died in August the same year. So they had her set up for 5 different sets of chemotherapy, and she was so weak that she wasn't able to complete more than 2. So it's much more difficult to treat in adults than it is in children. All right, this is showing myeloma with those additional lymphocytes. Anybody know what kind of white blood cell is involved with mononucleosis? It's not what you would think. It's not monocytes. It's lymphocytes. Okay, so it's an increased number of lymphocytes due to a viral infection with mononucleosis. All right, platelets. The megakaryocyte in platelets is so large that it can't leave through the sinusoids that are in the bone marrow. So here's bone marrow. Here's a developing eosinophil. The orange granules there. Here's another one. Here developing red blood cells that still have their nucleus. And here in the middle is this big megakaryocyte. That's what megakaryocyte means, is large cytoplasm. And it has a lot of infolding of its plasma membranes. It's like following a maze. And little sacks of membrane will pinch off. And they contain the chemicals that allow platelets to be sticky or to cause smooth muscle to constrict. And so when we look in the blood smear, it's these small granule structures, like this is a neutrophil, and these are the platelets. Right? So again, not really cells, but membrane sacs with those chemicals. Okay, let's take a break. And actually, you know what? Let's go ahead and do what I did this morning. Why don't you guys take about a 10-minute break, and then we'll have a lab period of about 45 minutes or so we did it 45 minutes to an hour this morning, and get the microscopes out. How many of you have used oil immersion?